Okay, so in this video, we're going to look at the topic of cell physiology, or CF for AS. Okay, so the first part of this, we look at movement of substances across the cell surface membrane, so across that phospholipid bilayer. First bit of this is looking at diffusion, or simple diffusion as it's called, and that is really when we talk about non-polar molecules. For example, oxygen, carbon dioxide can pass through the cell membrane without any protein molecules being involved. Okay, so they're very small, non-polar, non-charged, and diffusion is fine for those to pass across a membrane. Okay, here you can see diffusion in action, or diagram of diffusion in action. So we talk about if you put a drop of dye into a beaker of water, then that diffuses or spreads across the water or throughout the water, and the water is the dye color. Okay, when we think about diffusion across membranes, you need to talk about, or certainly think about, a number of factors in terms of the concentration gradient. So the difference in concentration from one side of the membrane to the other, the size of the molecule. If they're smaller, they'll diffuse faster. The temperature, again, if it's warmer, then the particles will move faster and diffuse quicker. If the membrane is thicker, then it will take a bit longer to pass through. If the surface area of the membrane is larger, then diffusion will occur at a faster rate as well. So just a little animation here in terms of different types of diffusion. So simple diffusion first.
In general, only small non-polar molecules can pass unaided through a phospholipid bilayer. Molecules move randomly. They tend to diffuse across the membrane from regions of high concentration to regions of low concentration. At equilibrium, equal numbers cross the membrane in both directions.
Okay, so you can see there, concentration gradient, large concentration or high concentration above the membrane here, lower concentration below. So there's a large gradient. You could call, let's say you call that 70%, and this is 10% down below. So there's a gradient there of 60%. So that's a large concentration gradient. As the molecules move through, you can see there the concentration gradient is maybe 50 up here and 40 down below. So there's only a 10% difference in the gradient. So it's lower. And then you get that equilibrium point is when it's sort of 50-50 on each side. Okay. And that's when you get no net movement. There's still movement going on, but the same amount would move up the way as move down the way. So it gives you that same gradient. Okay. In terms of facilitated diffusion, then, facilitated diffusion is slightly more complicated in that it requires a protein molecule. So they facilitate the movement of particles through the membrane. Okay. So with some of these, you have what we call channel proteins.
Some membrane proteins form channels that allow polar or charged substances to cross the membrane. In this example of facilitated diffusion, a channel allows specific ions to pass. These ion channels are gated and open and close by a variety of mechanisms. Here, a stimulus molecule causes the channel to open. In facilitated diffusion, like in simple diffusion, a substance moves from a region of higher concentration on one side of the membrane to a region of lower concentration on the other side. This movement down a concentration gradient is spontaneous and does not require energy.
Okay, so again, we're in terms of facilitated diffusion, we're thinking of higher concentration to low concentration, so down the concentration gradient. The only difference, I suppose, here is that these are polar molecules. They're not molecules that would pass through the membrane on their own because they're slightly charged. So they need this channel for the movement for them to be able to move through the membrane. Okay, often referred to as ion channels as well. You have these, they're gated channels in terms of they may need this little molecule here to allow them to open. That causes the channel to open, and then things can move freely down their concentration gradient again until equilibrium has been reached. Okay, and then if we look at carrier proteins, similar.
Some membrane proteins act as carriers that bind a molecule on one side of the membrane, change shape, and then deposit the molecule on the other side of the membrane. In this example of facilitated diffusion, a glucose carrier protein moves glucose down its concentration gradient, a process that requires no energy.
Okay, so again, we're talking here, it's a passive process, it doesn't require energy. High concentration again to low concentration, so down the concentration gradient. The only, I suppose, difference here is you can see that the protein, that carrier protein, actually changes shape as it moves molecules through. Okay, so that change in shape as the molecules move down the concentration gradient. Again, you'll end up with equilibrium, so same concentrations on either side. And you can see here you've got your carrier proteins, so that change of shape, allowing molecules to pass through from a high concentration to a low concentration. Here you've got your channel proteins, which open up and allow those ions, in this case, to pass through, again, high concentration to low concentration. The other type of movement that we talk about is active transport, and active means that it requires ATP. Okay, so you would have mitochondria somewhere around the area, somewhere within that cell that can produce the ATP, which allows then molecules to pass, in this case, against the concentration gradient. Okay, so they're moving from a low concentration to a higher concentration, and that's the bit that requires energy to move it against that concentration gradient. Okay, so ATP would be involved. It would break down, perhaps bind to the carrier protein, causes it to pick up that low concentration, whatever it is, glucose here you can see, it causes it to pick up and then causes the protein to change shape. So it's transported to the other side. You can see that in this little animation here. So you've got a high concentration of calcium at the top here, lower concentration at the bottom. ATP is used up to provide the energy to transport that against that concentration gradient. Okay, also seen in this little animation.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in a transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation. In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid. The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates, and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the 2 bound potassium ions dissociate from the protein and diffuse into the interior of the cell.
Okay, so the key differences between diffusion, either simple or facilitated diffusion, and active transport are that with active transport, substances are moved against the concentration gradient, so from a low concentration to a high concentration, and that requires, the second difference is that requires ATP, so it requires energy to be able to change the shape of or allow the protein to move them against that concentration gradient. Okay, another type of movement is cytosis. Okay, in terms of things moving in and out of cells. Okay, endocytosis is bringing things into a cell, so it could be a specific type of protein or smaller molecules like water, etc. And the way that it works is that this substance will pass into the cell, and part of the membrane invaginates, or it forms a membrane around that substance moving in. It sort of snibs off, if you like, and forms that vesicle around that substance that is passing into the cell. Okay, so that's endocytosis. Exocytosis is when material is leaving the cell. So this would be seen with those vesicles that nip off the Golgi apparatus. So there's modified proteins, etc., move to cell membrane, and the vesicle or the membrane forming around that substance will then bind to, if you like, the cell surface membrane. Okay, and it forms that continuous membrane around the cell, allowing those substances to pass out of the cell. Okay, two examples of that, or two types of endocytosis, if we take a look at, are phagocytosis, which is bringing in solid. Okay, so these would be solid, or penocytosis, which is bringing in a fluid or a liquid. Okay, think of Pinot Grigio as a wine, so that is bringing in a liquid. Penocytosis. Phagocytosis, bringing in solids. Okay, then there's a little animation that goes with that as well.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, penocytosis, and receptor-mediated endocytosis.
Don't worry about the last one.
If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis.
Solid.
If the material the cell takes in is liquid, it is called penocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clavin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell.
Don't worry about that one.
Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Okay, then we go on and look at osmosis. So osmosis is defined as the net movement of water from a high water concentration or a dilute solution to a lower water concentration or a more concentrated solution across a selectively permeable membrane. Okay, so water moving from where there's lots of it to where there's less of it. Okay, and you can see in the diagram here, we have the left hand, if this is a membrane, it's a partially permeable membrane, you see little gaps in between. You can see there's more water molecules on this side, so this would have a higher concentration or be a more dilute solution, whereas there's less water molecules, more sugar in this case, or solute molecules on this side, so it's more highly concentrated or less dilute. So water will move from the more, what we call a hypotonic, where there's lots of water on the left-hand side, to the hypertonic. Okay, lots of other stuff in it, so less, lower concentration of water, will move from left to right through that selectively permeable membrane until you get equilibrium. Okay, and you can see that here we have water molecules, and this should be blue but are green here. These smaller molecules can pass through the membrane. Okay, the larger molecules can't, so it's a more concentrated or hypertonic solution on this side, hypotonic solution on this side, so more water molecules will move from left to right. Okay, and you can see here, we talked about temperature earlier. If we cool this down, then the molecules are moving slower, they've got less kinetic energy, so diffusion or osmosis in this case would occur slower. And if we warm that up a bit, then they've got more kinetic energy, so the molecules will move faster. Okay, in terms of water molecules moving, you can see here pure water. Okay, and then if you put sugar molecules into that water, the polar water molecules, so those H2Os, they have slightly positive and negative charges, will be attracted to that solute or that sugar molecule that has moved or has been put into the water molecule, the pure water. Okay, and it forms something called hydration shells. You can see those little water molecules are attracted to that sugar, forming those hydration shells around, which may cause the sugar to dissolve, but it means that there's less free water molecules to move. Okay, so that has an effect on the amount of water molecules that can then move from one membrane to another. Okay, and that's seen in this little animation as well. So here you can see water molecules have kinetic energy, so they move around. Semi-permeable membrane here in the middle. In this case, we're adding salt, which is sodium chloride. You can see the water molecules forming hydration shells around those chloride and sodium ions, so that positively charged sodiums will attract some of those negatively charged oxygen molecules. The negatively charged chloride will attract the positively charged hydrogen ions or hydrogen molecules in water, and they form hydration shells. It also means that this is a hypotonic solution, hypertonic solution, so water will move from left to right, and you can see that level of water has increased. Okay, there you can see a sort of overview of how diffusion and active transport works. So simple diffusion, small non-polar molecules can just pass through the cell membrane. Oxygen, carbon dioxide, some water can pass through here. With facilitated diffusion, there's either channel proteins, so these would be polar, larger polar molecules that can't just pass through the membrane, either a channel protein, so high concentration to low concentration, or a carrier protein, which you can see changes shape there, high concentration again to low concentration. So that's facilitated diffusion. So the protein allows those molecules to move. Or active transport, you can see there is from a low concentration to a high concentration. Okay, so again, it will use a particular type of protein. ATP will be used up for that protein to be able to transport it against the concentration gradient. Okay, now as part of A level, we also talk about water potential within cells. So concentration is sort of replaced with the word potential. And water potential is made up of or is summarized in this equation that we can see the relationship between below. So the water potential of a particular cell, psi, is this little silly little y. So psi cell is the water potential of the cell, is a measure of the solute potential, psi s, plus the pressure potential, okay, which is psi p, okay, and it's a measure of the potential for free water molecules to move within a cell. Okay, and it's often shown as this graph. Okay, so here you can see that's the potential. The psi cell is the water potential here that you can see. You get psi p is the pressure potential, psi s is the solute potential. Now the highest water potential that a cell can get is 0. Okay, and that's when it's filled up with water. The pressure potential is very, very high there. Okay, so that's full turgor. A cell will be fully filled up with water, and the water potential of the cell would be 0. Okay, as the pressure potential drops or decreases, and you can see also that the water potential decreases as well. Okay, when the psi p or pressure potential equals 0, that's when your psi cell is pretty low here, pretty negative. When psi p is 0, we call that incipient plasmolysis. So that's when the cell membrane is just about to pull away from the cell wall, okay, and enter plasmolysis of a cell, okay. And at that point or below that point, then psi cell will equal psi s. So psi p is 0 along here when a cell will be fully plasmolysed, and psi cell equals whatever psi s is. And you might get some equations to work out in terms of what way water will move between cells. So it will move from a less negative water potential to a more negative water potential. Okay. The last thing they talk about then is osmosis in animal cells. So it's slightly different because animal cells have no cell wall. They, if water moves in, so if you put an animal cell, a red blood cell, for example, into pure water, it will burst, or it will lysis, it will lyse, because the water molecules will pass in by osmosis, cause the cell to fill up, no cell wall, so the cell will burst. Okay, in an isotonic solution, that's sort of the same as inside the cell. The same amount of water moves in as moves out. If you put an animal cell, for example, a red blood cell, into a very hypertonic solution, so something with lots of sugar or salt into it, then the higher water potential will be inside the cell, which will cause water to move out of your animal cell, and it shrivels up and looks a bit like this, or something that we call crenation. Okay, hopefully that helped with cell physiology.